Does anybody need to load their slides? Like last time?
Can I log in again?
How's your weekend, Rosie?
It was good. I got to go home. I was happy. I was on duty for most of it. So yeah. It wasn't bad.
It's pretty cool.
How was yours?
It's good. It's good. Got to fly a little bit. That was nice fun.
Out here?
Yeah. We're down by Palm Springs. It's pretty
Oh,
cool.
that's nice.
Yeah. Trying to learn a new airplane if I can. Scary. It's a lot of...
Is it big changes or like just like
This one was big. It was like, yeah, started. Yeah, it was fun. It's really cool.
That's so cool.
Okay, awesome. Thank you.
And there's all this.
Oh, hey, Liz.
How's it going?
Pretty good. Yourself? All right. How y'all doing? Nice to see everybody. All right, get rolling. Okay. So, if we remember where we were last class, we were going to kind of pick up from where we left off, I should say, out by about, I don't know, what were we, about halfway through your guys' group slides?
Just about.
Ish. Okay, so let's power through those on that part of the vision chapter, and then we'll jump into the slides. Does that sound like a pretty good plan? Now, did you guys on this side, did you guys have a chance to kind of work on your stuff a little bit more, tweak it a little bit? Yeah? All right, well, is it ready to go? Do you need to load it up on the computer? Or are there slides? Did you guys make any slides, or we're just going to go off the regular ones? Regular ones? Okay, that's cool. Sounds good. So we'll do that. I've had a request to make good on my earlier promises and be a man of my word by offering some neuroscience and beer. How you guys feel about that? We talked about this idea of neuroscience on tap before, right? So the idea is we get to hang out and talk shop, talk about the brain, talk about some psychology, maybe have a beer if you're thirsty, and if you're over 21, over at the campus pub. They won't serve you if you're underage, so don't try. Hang out, grab some chicken fingers or a burger, soda, water, iced tea, whatever. Does that sound pretty good? Yeah. Now, not everyone can make every single time, so I promise that not everyone's going to be happy, because it's impossible to make everyone happy with anything. Try to get your family to agree on like what to have for dinner one night or something like that. Even that's pretty tough sometimes. So everyone has different schedules. We'll do the best we can. I was thinking maybe Wednesday evening, maybe an hour or two after class. We get done around 4-ish. I don't know, maybe 5 or 6 or 7, or some time that you guys might want to give some feedback on. Might be a good time to meet up and grab a bite. Maybe a time when traffic is the worst, so you can hang out on campus while traffic is horrible, and then leave campus when traffic is much better. How many of you guys commute? Oh, just about everybody, huh? Okay, cool. What do you guys think about that? Does that day work for you, that evening, Wednesday evening this week, the 13th?
They have the STEM and psychology things.
What thing is that? And what time is it?
It's like 5 to 7.
Okay, so maybe that doesn't work so well. That's exactly what I'm trying to figure out. Great point. Okay, so do you want to do it later that night afterwards? Or just pick another day.
Well, it depends on what everybody else is up to. I mean, not everybody's going to go to the
Midterms this
Thing.
week. So.
There are midterms or no midterms?
Midterms.
There are.
Next week.
next week.
So next week? Okay. When do you suggest, so there's a lot of days next week. I mean, we try for every day, but, I mean, we're not alkies.
Do next Wednesday after exam or next Monday before.
You guys might want to kill me on that Wednesday, are you kidding?
Well, next Wednesday I'm just reminding that we have an exam.
But you guys want me to let you guys gang up on me after an exam?
Well, I think it would be good for us to all get
Review.
together and discuss what we missed and what we didn't miss so that we could all understand it.
That's a
better.
really good idea. Yeah, it's actually a really good point. So, we could shoot for that. Let me see. Do I? No. Nope. I have no life. Yeah. Juno's gone, so I'm not booked. And, we could do next Wednesday, the 20th. Does that work better?
Yeah.
Okay. So next one. Okay, so we started that off right. We found the right week. We found the right day. Now, what time is preferable for all you ladies and gentlemen?
After the test.
Of course, after the test. So certainly class goes from 2 to 4. So definitely after 4. I want to give a little bit of decompression time. You guys can like blow off some steam, curse me privately amongst your friends, and then come back and figure out maybe relax, hang out, do whatever you got to do. I don't know. Is 5 too early? You want to go 7? You want to go? I don't know. It's up to you guys, really. I have no life. My time is your time then.
5 to 7?
We could do that.
Can we bring like friends and children?
Oh, of course, yeah.
Okay.
Yeah, kids can't drink though.
Neither can I, so
Okay.
it's fine.
That works out good, yeah. Don't be filling those baby bottles up with the beer. That's no good. So yeah, definitely, I mean, it's open to the public. I mean, it's just hanging out on campus, so anybody who, if you have friends that, I don't know, think the brain is cool, or, I don't know. It'll just be basically hanging out, and we'll talk about the brain, and if you have crazy questions, or cool stuff, or, whatever. Talk about bring your neuroscience memes. So 5 to 7, what's that?
I mean, that works for me. I see some nods.
Okay.
Agree. What you think? That's it.
All right, 5 to 7, Wednesday. Next week, which is the
20th.
20th, 20. It's not mandatory, totally optional. If you don't want to come, whatever, do your thing. It's cool. But it's also seems to coincide with a good chance to do kind of like a test review. See where we went wrong.
At the pub?
Yeah. Yeah. Do you guys know where that is, by the way? Yeah. So it's is it San Manuel or Santos Manuel? San Manuel Student Center. Go up the stairs, and there's a little restaurant. So just hang out there. Sound pretty good? Okay. Yeah, we'll just kind of hang out. All right. Very cool. Did we need to go over anything else? I don't think so. I think I have everything on my list here. Okay. Anybody working on any cool neuroscience music videos that you're making? No? You are? Oh, man. Okay. Well, okay. Maybe you guys will get some inspiration at the pub. Anyways, okay, so we were talking about the visual system, right? Do you guys remember what spot on your slides that we had going on? We had, bring the screen up here. We were on. We ended off with the memes of winter is coming or something, right?
Down 2.
Down 2.
With the.
Oh, yeah, the illusions and all that. Lateral inhibition, right? Yeah?
Yes.
Okay. So, was it here? Wait, we did finish on this one. So, do you guys remember? Do you guys remember what the story was on this one? About how that lateral inhibition can create illusions sometimes, right? It creates and will exacerbate edges sometimes of luminance, right? As one is talking, it's shh, quieting down its neighbors so that its signal gets the signal's the same that it sends, right? But it seems like it's amplified because it tells the signals around it to go quieter, right? So it's a way that the nervous system kind of has biologically built in to make a cell's signal essentially louder or more prominent without having it change at all from what it's already sending, right? Because it's all kind of relative. And so if it's relative to what's going on around it, and you tell everything around it to quiet down, then your signal becomes heightened. And in this case, it allows a little bit of that extra, turns out, false perception, right? So our brains can play tricks on us then, right? We have illusions. Yeah, Rosie.
Okay, so these are the slides that I made, and I definitely captioned them wrong, maybe didn't read it right. So
That's okay.
A goes to A, and the 2 tiles that are right there are not the same color. That goes to the next slide's picture.
Yeah.
So sorry if that confused everybody. So just ignore the B part of it.
This one here?
The slide right after that picture. That's what the other part of the caption goes to.
Okay. You want to come up here and share it with us in a little bit more detail? Okay,
I
well,
mean, we did kind of go over a lot.
Yeah.
But
so we're starting off looking here, right?
yes.
So we orient everyone's attention here. And right, the cool thing about this, right? So like I was never actually a very big vision fan. Like, I like to see stuff, right? But like in terms of like neurobiology, I was always kind of like grumpy about it, and I kind of had a bad attitude as a student. And it's like, oh, I don't know, I just wasn't interested in it. I thought it was like, what am I ever going to need this for type thing. And then I think the more you think about it and dial in, it became really fascinating and really cool, because what we're starting off with here,
Yeah.
and as she'll share with us, remind me your name?
Columbo.
Okay. Is perception, right? And also reality. And it turns out our visual system, our brain will play tricks on us. So you'll have a difference between perception and reality, right? Because what we perceive is not always real, right? And we all experience lots of accidents of that accord in all of our lives, right? We think we heard something, it turned out to be different. We think we saw something and it turned out to be different. We think we experienced something and it turned out to be different, right? Any guys ever have those experiences? Yeah. Cognitive psychology is really fascinating for that. And these are kind of the biological bases for that. So we have, here's what we see, but it's also what's real, right? In the sense of there's a real stimulus here, and Columba will take it away and help us get to the next steps.
Okay, so on the book it tells you like cover it with your pinky, but obviously like it's a bigger screen, so I made another picture and I blocked it with like a black block, and then from here you could tell that they're actually the same shade. And then so how is it produced? I just like it's experience of our light versus dark, and it's created from the brain, and it just includes like the surrounding stimuli. And then the apparent brightness of the visual objects is subject to change by the visual system. And then when you cover the boundary between, it then equals an appearance that is equally dark, which is just shown in the picture.
Okay, so let's pretend we're at Neuroscience on Tap right now. Or maybe not at Neuroscience on Tap. Let's pretend that the whole class is not out drinking together. But now you're the only neuroscientist in the bar amongst your friends. You guys are just hanging out, right?
Yeah.
Maybe you're at Coachella Festival or something, right? And because people at Coachella like to have sort of philosophical conversations about the brain, right? Can you explain that to us in normal sort of parlance? like, how would you explain this to just a regular person, right?
Yeah.
Um,
I mean, isn't it just like shading and like the way you perceive it?
yeah, so what do we have? What is this accomplishing here?
So like when you're blocking the middle, it's balancing off the different types of shading.
So help us break it down, what's it?
The black block.
Okay, so this is balancing out what?
The middle line on the first one.
Okay, so our illusion is focusing upon this?
Yes.
Okay, now, is the idea then that this gray is the same as this gray?
Yes.
And is this gray the same as this one?
They should.
Yeah, okay, yeah, we're assuming that that's created at the same colors scale, right? And so, what is the point of this illusion that it's getting to? Is it's about these issues in here, these, right? There's a little bit of black, there's a little bit of white, and then it kind of fades, right? Is that what we should be focusing on here? Is that the part of this illusion?
I mean, isn't it like the way our brain receives or like perceives those shading?
Yeah, so here's another thing, right? So let's say you're hanging out at Coachella, you need to show this to somebody. You say, look, do you see shaded colors here? How many of you guys see shaded colors? Yeah. And are they really shaded?
Looks like white and one different versus the other.
So we talk about things being equally dark, right?
Yes.
And they reflect the same amount of light. So how is this produced, this appearance that they're equally dark?
Coming from our brain?
Yeah, so what do we mean by our brain, right? That's a big, huge lump of tissue.
Yeah.
What's the process that we've been talking about, this lateral inhibition, right?
Yes.
It's kind of what we were talking about then, right? So you're gonna have that neighbors quieted down so that the main signal can go forward. And so, if you block this out, don't these look basically the same? That's what's real. But what you see and perceive are these extra shadings. It's sort of a simple letter. Level, that is. How do you guys feel about that? Yeah.
Could you actually just run through lateral inhibition again?
Let's see here. Do you guys have stuff on lateral inhibition coming up?
It might help on the next slide, I'm
Okay.
not sure.
Okay, let's do that. Oh yeah, there we go. Help us out. Go.
Okay, so it's like showing us here that, like what he was saying, like when it quiets down, it's blocking the other ones.
So let's slow it down a little bit. And I'm gonna get kind of pokey on you here. Okay? What's it?
The receptors.
Okay, that's what I want to be talking about. Okay, let's make sure we clarify, right? Because you know what it's like to be a student, right? And if I'm just out here talking, you don't know what I'm always talking about, unless we're really clear about saying that. Let's try it again.
Okay, so since they're shown here 4 receptor cells, the ones in the middle are going to be the ones that are more prominent since the other ones are being quieted down, which is the dark and the light.
These are our receptors. I'll be your little assistant. You guys still know what Wheel of Fortune is? They still have that? It's probably played on like a Angry Birds app or something now, right? Something on the television. Do they still have the TV show?
Yes.
Okay. All right, so I'll be Vanna White for her. Right?
Okay, and then when I was confused, I couldn't really know, but like the graph part, since I know it relates to it, but it's like I'm not sure like what it means by it. Like,
Yeah, good question. So this is what we finished off talking about last class. Do you guys remember? Okay. And so this is, I'll tell you what, I'm going to make it a little bit bigger. Let's see here. Okay. Is that easier to see? Okay, because we're gonna read some of these points. So remember we talked about breaking down slides with the axes of the graph, right? So start, especially if you don't know what it's showing, and you're like, oh geez, what's it showing? Right? Help yourself. right? Start walking it through, because as you're showing it to the audience, you're also helping yourself. You're going to say, on the x-axis, we have the position of where the ganglion cells are. That means, is it here, 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 or here? So this dot is this cell, this dot is this cell, this dot is this one, and here is here, right? Now, here's the thing, right? It's where the cell is. And the y-axis, that is where on this part of the graph it is basically how much neural firing there is, the rate of firing of those cells, okay? So, it's these ganglion cells firing. So this is what's detecting our light. This is what is signaling about the light to the brain, right? So it's kind of like the pitcher, and this is the catcher, right? So this is, well, that's kind of a bad analogy, but like they're sending the signal like the pitcher sends a baseball to the catcher here. And the catcher is sending the signal out. I don't know, the only way I can make the example work is if the catcher throws the ball to the second baseman to throw someone stealing out at second base, but don't belabor that example too much. I was just trying to roll with it. Anyway, so what we have here is this is how much firing is happening for each of these yellow cells. Now, this is what the stimuli is. These two cells are receiving dark, and these two cells are receiving input of light. Right? So, if you rotate this a little bit opposite, like say 90 degrees, so instead of being left and right, it's dark and then light, then you have what we were looking at a few slides ago, those boxes. Right? So you have a thing, you had the top part of the box and the bottom part of the box. We had that illusion in the middle. Remember? This is the same thing, except that now it's gone this way. So the dark and light. And this signals are coming down here, and this is going to show us some information. Who remembers what I was talking about with this at the end of class last time? Can anybody finish my sentences for me now? All right, we'll review it. So, check it out. What's going to happen is we're talking about the lateral inhibition, right? So let's just keep it real simple with this one cell. It's in the dark. So it's always releasing glutamate because it's depolarized in the dark, right? So as this is in the dark, it's releasing glutamate here, which means this cell is firing. It's firing a positive signal activating this retinal ganglion cell. Okay? Okay, so this is activating that, but it's also sending an inhibitory signal telling its neighbor, shh. Okay? So, as it's doing that, okay, this is about where it's at. I should say, yeah. So, let's move one spot over. Still in the dark, right? So, it's not, these retinal ganglion cells won't fire because it's in the dark. That's why this is nice and low. However, this cell is also sending, this, basically it's firing here and it's going to have an inhibitory effect on this cell. So when it does that, right, it's also going to send this cell, shh, right? So this cell is already going to be quiet. Basically, these retinal ganglion cells are going to be quiet in the dark, right? Since it's quiet in the dark, it's going to not have a very high signal. Well, it will. It's going to send this other inhibition here. Okay? However, now the light is on here. Okay? This cell is going to send its signal to this one now, right? It's going to reduce its firing, which means this is going to be activated more. So now you're going to have this inhibition over here. And because this is sending more inhibition, now the cell is activated, right? It's telling this cell to be quieter, which is why it goes lower than this one. Does that make sense? Yeah.
No, can you repeat that, please?
Yeah. So, out of these two cells, which one is really getting told to be quiet? The second one, right? Okay, why is it being told to be quiet more? Because this cell is being activated. When this cell is activated, this line is activated, this is going to send a bigger signal to be quiet here because it's being activated. Right? Because this is being activated because of light, it sends this to be quieter. So it's going to be a little bit, see how it goes down a little bit? Okay. Does that help? A little bit?
Yeah.
Okay, yeah.
So then the reason why it's quieter is not so much because of the left receptor cell, but so much on the right receptor cell, the one that's in light.
Yeah, but of course it's relative, but yeah.
Right.
Yeah. Yeah. Correct. Yeah. I don't want to say right because there's rights and left cells,
Yes.
but yeah. Yes, correct. Now let's move to this side, right? So now this cell is activated, and so is this one because they're both receiving light coming down. Okay? We start on this side. This is sort of the normal level, right? That's what it's kind of getting activated with. And let's see, so it's going to send a signal here, it's going to inhibit this one, but this cell is also sending an inhibitory signal here, right? Now, here, this one here, see how it's a little higher than this one? Right? Okay, so this is a little bit higher. Why is this higher? Well, the reason this is the same is because this one's lower. It's just same but different, right? So this cell is going to be firing a little bit more because it is receiving inhibition from this neighbor, right? However, is it receiving inhibition from this neighbor? No. So if it's less inhibited, is it going to fire more or less? Just a baby bit more. So this one ends up firing a little bit more. This one ends up firing a little bit less. I should say these retinal ganglion cells. And you end up with these slightly different illusions. That is slightly different. Basically, we're not robots, said another way, right? If every time there's light, the cell has to respond in exactly the same way to show the exact same amount of light is happening, that'd be kind of robotic, right? It kind of works like that, but because there's these factors, we get this thing that's kind of not. So robots really can't have illusions, can they? It'd be kind of weird if they did. If you think about it, right? The robot's a machine. It sees exactly, this is the light, whatever, right? So, yeah. We're not robots, and our cells are going to respond differently based on the influence of the other cells around it, giving us that illusion. Just a little baby illusion, no big deal. But we can see there's that exacerbation, and that's going to give us a better detection of that border. We're going to see that exacerbation a little bit. Does that kind of jive with what you were studying?
Yeah.
Yeah? So, two central receptor cells differ more in their rates of firing than do either of the two left red cells. Because of these lateral inhibitory connections. Right? So what you can do is imagine that there's no purple cells here. Is this graph possible? No, why? What would it look like if we had no purple cells here, no bipolar cells? Yeah, it'd be a flat thing here, and then another flat thing here. Right? You wouldn't have these little extra parts. It would just be robotic. It'd be like, because you would just have, stay in your lane. Kind of like car lanes. So imagine, wouldn't the highway be really cool if when you were driving you had like a cool little video controller that you could like, you know how, you guys ever play Mario Kart? Okay. Yeah, now we can talk on the same level. Okay. So Mario Kart, you know how you're like cruising around there and you've like collected these bananas and stuff? You can fire off a couple bananas and you leave them there because you're racing the other people on the highway, on the racetrack. And if you shoot those bananas off just right and you're really lucky, then your competitor racing on the street will snag that banana and go woo and then flip out. Right? It kind of works like that, right? Do they still make those noises in the game? Okay, cool. It's been about 15 years since I played, so I was hoping I didn't just randomly pull out a weird noise. So that's kind of what imagine that these are lanes, like car lanes on the highway, like a 4-lane highway. Wouldn't it be cool if, like Mario Kart, you had a little controller in your car where like you really need to get somewhere and there's some total jerk off in the lanes next to you, either slowing down or trying to cut over, and you can like send a little signal over that slows them down and allows you to keep going further? That'd be awesome.
I kind of have that
You do?
in the newer games, yeah.
Oh, I thought you meant in your car.
I don't know. I'm still in Mario Kart.
Yeah, well that's what the bananas are. In Mario Kart. Yeah.
Everyone's like, whoa, what?
Yeah, can I have your car? They make those now? Wow.
Anyway.
Yeah, right, so that's what I was getting towards with the bananas in the Mario Kart, right? They're the things that you shoot over the side and slow them down. Or whatever, the little ink blots or whatever. But wouldn't it be cool if you had that in the real highway? That's what lateral inhibition is. You're inhibiting your lateral neighbors so that your signal can go further. Does that help kind of connect the dots a little bit? Right, so these are your lanes. And so you want to send that signal over here to keep this quiet, which is going to allow this to go further. Now, this is what's actually activated. So I should be focusing on this one. So here, the signal's coming here. Because it's activated, it allows you to send that banana peel over here to slow down this car in the lane next to you, which allows that to go just, imagine this is not neural fire, imagine this is speed. Okay? This is going just a little bit slower than this one is now, isn't it? Does that help a little bit more? Think about it as speed in car lanes, right? And because this is getting the gasoline, right? Your light, you got a full tank of gas over here, the light is kicking it up. It's going to be going much faster, right? So, and because this one is getting activated by the light, it's going to send that banana peel over here to calm down its next lane. Yeah, so.
Well, on the graph, the way I'm reading it, it's not over time the rate of neural firing, it's just the position of the ganglion cells. So when they meet in the middle, that straight line down, does it serve, like what purpose does it serve? Is it just indicating that that's the point in the middle? Because it doesn't say that because this is all happening simultaneously, it's not like one at a time. So then it's just indicating that that's just the middle point.
Yeah, that's a really good point and observation. If anybody didn't catch what Lisette was asking, she's like, okay, well I get this, I get this, I get this, and I get this, but what the heck is this? And honestly, it doesn't represent anything other than that's just the transition point. To my reading of it. Now, heck, I might be wrong. Golgi was. He's got a Nobel Prize. I mean, Aristotle was wrong too. He thought the brain was a radiator, so it's okay to be wrong. We're in good company, right? But my understanding of this is that, right, this is kind of like lots of line, you guys do math with line graphs, you make a line graph whenever you're plotting something. I don't know. I do a lot of data in my lab, so I do lots of line graphs, but I don't know how much you guys use those. But let's say you're making a line graph. The lines are just connecting spots. The dots are the data. That's what this is. Data, 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 because it's 1, 2, 3, 4. This vertical line is just
It just indicates the point where they transition, but it has no other.
Yeah. Because so right, this is a cartoon, right? So it's just theoretical. It's just a model, right? So what you would imagine is there's actually a ton more cells packed in here. So the line represents theoretically all those other cells. So yeah, they'd be getting more and more lesser over here. You see what I'm saying? Really, really good, deep reading of that graph. That's what you should be doing. You should be picking up that graph and dialing into exactly what every single element means, so that then you can ask, well, what does this other thing mean? And find like, oh yeah, it's kind of just part of the cartoon that doesn't actually make sense. I used to do that in grad school because I was having so much struggle trying to understand things in the class and the slides and stuff, and these new classes I was taking that were really tough, like molecular biology and genetics and stuff. And I would go to the professor's office all the time, and I'd have spent like an hour looking at this, trying to figure out what the heck this whole thing meant. And I'd go and I'd ask a question like that, and they'd look at it and be like, "Yeah, you're right. I never thought about that. The figure's wrong because it shows." Yeah, because like the figure's made by experts who like think past that, right? They're trying to show a completely different topic or a different point, right? And they're not starting at a beginner spot where we're actually look at it and I'm like, I want to understand this. Let's really understand it and break it down. And then you get to the point that made sense to the expert because they were like, of course, you know, da-da-da-da-da. And they publish it because the other experts who review it also have the same bias and they just overlook it. And then they sell it to a bunch of students who are new and we're like, what the heck does that mean? So yeah, I mean, that's exactly what you ought to be doing. Yeah, it's really good. All right, Columba, what do we got next?
No, next one's just a meme.
Oh, even better. All right. What's the other one? OPPA style. I like that. That's good. What's OPPA?
It's just a Korean word for oppa.
Opa. Isn't that Greek?
No, you know the song, the Oppa Gangnam Style. Sing it.
Oh, yeah. How does it go?
I thought you were gonna go for it.
I forgot.
Yeah, no, I know Gangnam Style. I just didn't know that Opa was a part of it. That's cool. You guys know, in Greek, it's like Opa when they're dancing. Like, Opa. I was thinking that. Actually, what I was thinking, I didn't see the A. I thought it was OPP. Like, you know me. Yeah. That's how old I am. I remember when that song was cool. So, all right, that's cool. Good job. Let's see what else we got next. All right, Sean K. Let's do it.
We kind of go like back and forth because the
Sure.
picture goes with the slide.
Yeah, you can control the mouse if you want, or you can just tell me what to do.
Oh, yeah, I can do that and like go back and forth.
Have the floor. The back button. Just roll it forward.
Okay. So the ganglion cells pass through right. Is there all there? Yeah. Pass through right around here, through the optic canal.
Is it canal?
Where that's the opening in the skull, where they pass through.
Okay. I thought you were talking about the other word you have,
Oh.
chiasm.
No, that's back.
Okay.
Up here, yeah, up here, optic canal, the opening in the skull where the ganglion cells pass through. The temporal or outer retinas, which is blue on this one, red on this one. They cross at the optic chiasm.
Chiasm. You want to see a trick?
Okay.
I'll show you something here. Snag that, or actually, we'll do. Copy that. Put this here. It's gonna come over. And where's font size?
Next, yeah.
Very good.
Works.
No, you don't have to roll. Well, I tried.
I don't know.
Now keep it open.
Okay, so optic chasm. I couldn't really find why
Why what?
they cross to,
They cross at the chiasm?
yeah,
Well, okay, so that's a good question. So, Sean, is it right?
yes.
Okay, so Sean is asking, why do they cross at the chiasm? Now, I get a little wonky and nerdy in some of my terminologies you guys probably know, right? So in terms of why, I don't know that, I don't look at it like we can know an answer to why they do, because like basically, I wasn't there chilling with God when he made it, right? Like, what we can say though as scientists, right, is we can observe some advantages of why it seems to be useful that it does that.
Okay, I had an idea. That,
Hit, what you got?
you sustained some kind of injury to one side of your brain, you'd still have a pretty decent field of vision.
Yeah, so a redundancy system, right? So, I mean, he didn't give us one leg, he gave us two. I mean, mostly, right? Congenital situations notwithstanding, right? You guys ever seen the movie 300? Right? You remember King Leonidas and his brave 300?
This is Sparta.
Yeah, this is Sparta. Bunch of airbrush dudes and jacked up six packs. Right? Okay. At the end of the movie, they send one guy back from the 300 who died at the Battle of Thermopylae, which is a real battle, by the way. It really happened in ancient Greeks versus Persia. They sent one guy back. Remember why they sent him back? Well, there's two reasons why they kind of sent him back. You guys remember? He was blinded in one eye because he got hurt in the battle, right? He also sent him back because he had this great additional skill as a warrior, that he was also a poet and a writer, which they're like, dude, we need somebody to tell our story, right? So he said, yeah, but anyway, so the point is that he lost an eye, and and the guy said, are you all right, man? You lost an eye. You guys remember what he said? He said, yeah, but God saw fit to give me a second. All right, so he had a second eye, redundancy system. Because horrible things happen in nature, and we need extras. So you have this information. So what Sean was talking about was, first you start with the eyeballs on, we're looking at the eyeball right from a top bird's eye view down, and you divide the retina in half, like we talked about last class. So half of a retina we call a hemiretina. Wherever that is, it's over here somewhere, hemiretina. And you can name the left and the right side of it, right? Except if I said the left eyeball is left hemiretina, and the left right hemi, that would be so confusing. So they can't use left and right of here. They call it nasal, because it's by the nose, and temporal, because it's a little further away. So you have the left temporal half of the retina, or left temporal hemiretina, or the left nasal hemiretina. Good way to start things off? Okay, so you have that on both sides, and then, like Sean was saying, they're both going to send their information into the brain through which optic nerve? Well, through the optic nerve.
Yeah.
Crap, that's a bad question. Which cranial nerve, right? Yeah, okay, so, hey man, at least I don't think the brain's a radiator. Okay? We all got a leg up on Aristotle. And the idea is that it's going to send both of those lines of information in, and we're going to see that one stays on the same side of the brain, and one crosses. What was the name of that cross?
Optic chiasm.
Chiasm. So chiasm is cross in Greek. I know you guys probably aren't. You guys probably don't remember all your Greek classes, right? Okay. But yeah, so chiasm is it means X or cross. Right? So pretty easy if you ever need to remember it. How many of you guys are in sororities or frats? You guys know the Greek alphabet, chi? What is it? Isn't it a cross? X?
Yeah.
What's that X on your shirt?
Chi.
Yeah, chi. Chiasm. Cross. So you're going to have this information, like Sean was saying, coming in a redundancy system. And it's going to come to the different sides. And what it's showing, particularly, is more about the field of view, right? We have the visual field. So this part will go through will capture anything from essentially here. So anything in purple is seen by both eyes. Why is that true in purple? You guys remember guys? You guys remember kindergarten art class?
Doesn't red mean?
Oh. There's gonna be different information,
Yeah, no. Yeah, so that's actually really helpful, I find, because it lets you know that the authors and the illustrators are working hard to actually make the figures make sense if you really dig into it. That there's like a method to the madness. It's not like they're more like the kindergarten teacher than the student. Like the students in there they're not just coloring randomly, filling the triangles in. They're actually making it systematic. So everything in blue is seen by these blue sides of the eyes. Everything in red is seen by these red sides of the eyes, the nasal and the temporal. Right? And of course, we can see the areas where they overlap. And anyways.
And then binocular depth perception is pretty straightforward. It's just that everything you look at, even if it's just by fractions of a millimeter, is a different distance from each eye. So that gives us the depth or three-dimensional vision.
Okay. So let's talk about some other. What does it mean to be ipsilateral and contralateral for this visual field? Yeah. So ipsilateral is the same side. Contralateral is the opposite side. So what's an example of ipsilateral processing here?
So is the temporal view on the left eye?
Well,
And in the contralateral view, the same field of vision, but shown in the right eye.
okay.
Would it be what's seen by both eyes? Or no?
Huh?
Would it be like the field of view that's like shown on both sides? Will it be ipsilateral or not?
Well, I was kind of thinking about it in a different way. So, what part of the visual field ends up there?
The right.
The right. And what hemisphere of the occipital cortex is this?
Left.
So would we consider that contralateral? Is there anything that we can see here of the visual field that's processed ipsilaterally? No, it's kind of tricky, huh? Looks like it almost had me too. I almost forget this at times. It looks like this is processed ipsilaterally. The blue is here, but what is the blue actually showing?
The right field.
The right field of view. So it looks like anatomically, yeah, you have, if the question is, right, questions matter, right? I hope none of you guys are ever accused of a crime and have to be a defendant in a court case and have your future rest upon what some witness stand person says. Because if that does happen to you, then you want to make sure that those questions asked to that person are perfect, right? You don't need the question to be slightly wrong, because then they're going to interpret it wrong, and you're going to get the wrong information. So if the question is about the anatomical part of the eyeball, the retina, then yes, that side that's processed ipsilaterally here. But it's also processed contralaterally there. But usually on the tests, I say usually because I'm just going off my memory because I don't have the test in front of me. Usually people are asked about the visual field processing in the anatomical locations. Do you guys see how that's a really different question? Yeah? It's also a really different answer.
Always contralateral.
Yeah. And, yeah, so, anyways, we have that. What else you got for us?
I had 2 slides, but now they're 1. So.
Oh, that's even better. So more efficient, yeah.
So does it become ipsilateral if they do the thing where they split the brain in half to stop seizures?
Ah, you mean split brain colosotomy.
Yeah.
Just cut the corpus callosum, right? It's an interesting question. We'll get into the visual attention system a little bit. Well, actually, I don't even know if we're going to get to that chapter, but so you get these weird situations about the ipsilateral and contralateral visual field access. So what happened? So, right, so here, cartoons are just cartoons, right? So what we're seeing here is this cartoon just shows the blue color stops and the red color stops. How many of you guys think that that's just vision, that just stops there, we're done, game over? No. I mean, you guys have had biopsych, whatever, you know there's more to the story, right? Okay, so, from here, right, it's going to get projected all sorts of other places, and it's going to start crossing at the corpus callosum. We're going to get higher level integration of this stuff, right? Which is also still partially kind of an existing mystery. Like, we still want to understand the bigger picture of how do we see and integrate across our hemispheres, across our visual field. Where does this cognition happen, right? It's a deeper question that people are still working on solutions for. So the corpus callosum helps those hemispheres to talk with that information at higher level integration. When you cut it, people can have really weird deficits of not being able to see parts of the other visual field. You guys want to hear the wild part about it? Like, let's say, I don't know, let's say he had his corpus callosum cut, right? And just to keep the example really simple, let's say that he couldn't see stuff here, like wherever my hand was with my pen, right? And if you ask him what's over there, and he's like, nothing, right? And if I did this, he'd catch it.
Oh. I thought I was not supposed to see it.
No, that's okay. But you see how he still tried to do something? If he couldn't see it, his hand would have stayed still.
Is that something to do with the spatial ability and the language on the left? Spatial ability?
Not so much language, but
Well, depending on the lobe that you're referring to, right?
yeah, but not so much language. It's more of a visual attention thing. It's called. You can also think of it as hemispatial neglect. It's is another type of clinical condition and stuff like that. When you have brain damage to some cortical areas too. But the idea to your question about cutting the corpus callosum, yeah, you can have these weird deficits where you can't see certain stuff in certain visual fields because it won't allow that crosstalk and integration. But yet sometimes you can have like cortical blind sight, is another term you could hear, where you can have damage to the cortex and you can't see a certain part of your visual field, but yet you know, you can, I don't know, they throw a tennis ball at you, you might grab it just as a reflex. So something is the information somehow still getting in, somehow weird, wacky way. It's not reaching your consciousness threshold. But yet it's in there for you to defend yourself against a tennis ball or a Nerf ball, right?
I was gonna say, there's a video on YouTube of a guy, he's blind, but they had him walk down a hallway with different obstacles, and he walks around them. But as far as he knows, he's blind.
Yeah. Yeah, I mean, it's pretty wild. Yes, man.
Is that how, I don't know, it's with all the Robert De Niro, and he's in like a coma, I think?
You're talking about the movie Awakenings.
Yes.
Yeah.
And how like they figured out that like mental patients who like weren't like really aware of like what they were doing, they threw a test at them and they were able to catch it.
Yes. However, remember we talked about perception and reality? We also talked about all sorts of memory wackiness in class and how it's not always right. This is a really good example, right? So I primed you with this tennis ball thing, right? So that's priming. And you were able to remember this movie scene. Here's the problem, I think. And either you had it wrong or I had it wrong. But one of us has it wrong. In the movie scene, it wasn't a tennis ball. Pretty sure it was a real baseball. Just like, ouch. So you got a patient. He seemed like he was in a weird catatonic. You guys know what catatonia is? It's like you're just kind of, you're not in a coma where like sleeping, but you're like frozen stiff, but you're awake, but you're unresponsive. That's like catatonic. And he was that way. He was in a wheelchair. It was Robert De Niro. You guys know Robert De Niro from Meet the Fockers? Okay. But it wasn't a coma. He was this it seemed like paralysis from, it turns out, Parkinson's disease. So Parkinson's disease, when we get to motor control sections of the class, we'll talk about movement, right? So if you can't move, you're paralyzed, right? But he didn't have a neck injury, didn't have a back injury, he wasn't a paraplegic, he just couldn't physically move. And it's a true story, actually. What's the guy's name? Robin Williams plays the doctor who makes this genius discovery. He realizes that if he threw this baseball, now he wasn't walking around the mental ward throwing baseballs at, catatonic patients. That would have been, like he wasn't a jerk. I forget the storyline of how it came about, but he realized that he did not lack the ability to move. He lacked the ability to have motor control on a voluntary level. He did not lack it on an involuntary level. So if you threw a baseball at him, he'd be like, and Robin Williams is like, holy shit, you just moved your arm. Like, you're not supposed to be able to move. How the heck did that happen? And he traced it down and did some studies. I'll get you in a second, Seth. And it turns out he was able to discover a preliminary treatment for Parkinson's that hasn't totally worked, but I should say this better. He discovered that dopamine chemicals were at the basis of some Parkinson's disease because he made a lot of dopamine treatments for delivery to that. And he turned out that the guy, when you jacked up his system with this huge dopamine cocktail, anybody, do you remember what he did? The patient, De Niro. When he got the treatment? He got up, he started dancing, running around, he broke out of the asylum, went to go chase some girls around town, went to go live his life. Yeah, wouldn't you? And there's a whole other story, really recommend it. We should do like a movie night one night. I say that to my lab too, I have a lot of movie nights. By the way, speaking of movie night, next Monday, I'll be doing a movie night with the Residents Hall Association. Basically, and you guys know Coyote Village? It's a big dorm, brand new, really swanky dorm. Big, huge sort of room in there, big screen. We do a movie night on a space movie. Hang out about 8 o'clock, Coyote Village, multi-purpose room. We're not allowed to advertise which movie because apparently there's some sort of copyright infringement. So I just say it's going to be a space movie with Matthew McConaughey. Does he do stuff over Scott? I didn't know.
So you said Matthew McConaughey?
Yeah.
Oh, you're talking about Jake Gyllenhaal. What is
Oh, sorry. Sorry. So it turns out it can be two options, right? So he's in Interstellar. He's also in this movie from the 90s called Contact with Jodie Foster. It's a really good movie. So I was going to do the old school one because I know most of you guys probably don't know the old school movie, but it's actually a really good movie and it's kind of ahead of its time. And you get to see a really young version of him. Anyways, we'll figure out that one on site. So 8 o'clock, you guys want to come. There'll be food. So anyways, enough about movies. We'll get to Awakenings and maybe that when it comes from the motor system. But we have the visual system, and we have this visual field. We can talk about cortical blind sight. We talk about hemispatial neglect. Talked about cutting the corpus callosum. Do you got anything else for us? Good job, Sean. Okay, so what do we got next? Who's up next? Is this the same thing? What do we got here for VR?
That was great.
They might not be here. I don't know.
They might not be here? Okay. That's kind of cool. So, anyways, the idea here is we've got these are the same things. You have visual fields, right? But see how it's the same idea? But now the eyeballs move to the side. Right? It's just a different animal. It's a rabbit, right? So animals of prey, they need to see more, almost 360 degrees all around them, so they don't get eaten, right? Kind of like, you guys remember when Pumbaa was getting chased by Nala? Pumbaa, yeah, you know? Yeah, Pumbaa's getting chased by Nala, and Timon's like, what's going on? And he's like, she's gonna eat me! You guys remember that scene from The Lion King?
Yes.
Anyways, that's because animals of prey don't want to be eaten. So it helps them evolutionarily to have eyes that can see much more around them. Whereas things like you and I are predators, right? We want to eat some delicious bacon, which means we got to catch that pig, Pumbaa as it happens to be, and so we got to look straight ahead. So our eyeballs are straight. Anyways, okay, so that's kind of what's that showing that here. What do we got here for you guys? People not here? Okay, so let's take a look at what we got in this pathway. Now you guys stop me if I'm covering stuff that you guys are slated to cover in a little bit or somebody else. But what this is showing is that basic pathway from the eyeball to the cortex and then beyond. So we talked about this idea that the information goes right from the, let's start, we did the molecular cascade, right? From rhodopsin to transducin to PDE to cyclic GMP gated sodium channels. All right, hopefully you guys have that written down, that this sequence. All right? So we have then the photoreceptors, the bipolar cells, the ganglion cells. Of course, you've got the lateral inhibition from the horizontal and amacrine cells, but we've gone from the retinal ganglion cells now, sending those action potentials through the optic nerve, the optic chiasm, the optic tract. So now we're coming here through the optic nerve, the optic tract, go through the chiasm, here crosses. It's going to go, its next destination, its next big destination is the thalamus. Okay, so that's kind of over here. Now it will send a little bit of an offshoot down to this midbrain area, okay, called the superior colliculus. And the guy who taught me that class in grad school about the superior colliculus had a very thick German accent. He's a very nice guy, but I can't ever think of superior colliculus without having it said in an accent of like, superior colliculus. And so I always think about that. And so you got the superior colliculus, but really the main destination is the thalamus. Okay? Now you guys probably all learned the thalamus in your earlier classes. What we're going to do is we're going to dive in a little bit deeper. And it turns out the thalamus is chopped up, sliced and diced into a ton of different subnuclei. That is little baby nuclei within this structure of the thalamus. Now for vision, there's a really famous part of the thalamus that is this stopover point for the visual information. And you'd be totally good if all you call it is the LGN. LGN, and it stands for lateral geniculate nucleus. Okay? So I'm sure this is, yeah, there it is. Yeah, there. So that's about halfway in our processing stream. This is our thalamus. And from the thalamus, it's going to get sent to the cortex, particularly the occipital lobe of that cortex. And it's going to get sent there by something called optic radiations. So they're projections, thalamocortical projections. You have neurons in the thalamus send their axons all the way out to the visual cortex. Those are called thalamocortical projections, because they're projecting from the thalamus to the cortex. Sounds like big words, but it makes sense. It's actually pretty descriptive and just tells you exactly what it is. And so that's kind of what we're showing here. And this kind of shows that same information, just shown in a different picture. Now, like I said, you guys stop me if I'm covering your ground. If somebody else wants to come up here and we get these slides. Is this something
It
you?
might be for the next 4 more slides.
Okay,
That person
that's cool.
wasn't here.
So this is the same information, right? Optic chiasm, LGN, optic radiations to here, right? This is the medial view of the occipital cortex, medial view of the other occipital cortex. This side's on the left hemisphere, this is the medial view of the right hemisphere. Okay, medial means on the inside. So what is the value of that color, of all those colors? Were they just bored? Did they just want to make a rainbow brain? Why did they put those colors there? Anybody have ideas? Anybody thought about this figure?
Because like when you're looking at something, you see more than just like red or blue, purple. There are other points also that like your eye has to take in.
Yes, although I would steer that away from the interpretation of color perception. All these different colors are not about the colors you see at all. Yes, sir.
That particular picture picture just represents more of a three-dimensional view of how the site works.
Yeah, well, it is kind of a three-dimensional illustration of it. Yes, ma'am.
It's the side of the brain process, what you're seeing.
Yeah, so we're getting hot on the trail now. So what these colors are useful for is to show you what part of the visual field. So everything Sean started talking about, right? How we cut this visual field. So he just talked about 3 slices, right? What you looked at as red, blue, and then purple in the middle. Purple was the overlap. Now we're actually looking at this. So here we have red, blue, and purple again, right? But we've got some, what this is able to show you is the different visual fields of each eyeball and how and where those areas project contralaterally in the brain. Okay, so let's break this down a little bit. These dotted lines are really important. See how this, they show you that this is the visual field of the left eyeball. Now, what they could do to make this even a more helpful figure is if they printed this twice, and in each one they only showed one visual field, rather than it's overlapping, because to me this is pretty confusing. Yes?
By reading it, essentially the outer side on my right, so essentially the dimmer colors, yeah, does that just indicate the peripheral view? Whereas once they're overlapped, that's just what both eyes can see then?
That's a really good point, Lisette. You can help to read this by the deeper, richer colors versus the fainter, lighter colors. So the fainter, lighter colors means that's where only one eyeball can see. The places where it gets a deeper blue versus this lighter blue means that both eyes can see that. So you have 2 light versions of blue overlap, so it looks darker. Does that help? So we can see there's a big chunk in the middle of our field of view that both eyeballs can see, which you know already because you just cover up one of your eyes and you see just about everything still, right? So, because each eye sees just about the same thing in the middle. Now what these crosshairs are useful for showing, right, is the information that goes to the hemiretina sections. Nasal, temporal, but also vertically, the top half and the bottom half. That's why you've got this crosshair. Now these lighter fields here and here are indeed the periphery. The stuff that only one eyeball can see that you're going to lose if you happen to lose it, right? And what you can see is this. So here, this is really useful. Just look at purple. See how purple's on the bottom here? Remember we said that the lens inverts the image on the back of the retina? So it goes through the lens in the eyeball here, and purple is now on top. But it's on the bottom here. WTF? Yeah? Question? Okay. So see how this got inverted? The bottom part is on the top part here? And let's see. See how it's also on the outside? Look, purple here is on the inside. What the heck? That's what these colors are really useful for. So you get these colors, they're mapped, it shows you these different sections of the visual field, how they're mapped onto different parts of the retina, how from the different cells in the retina, the different retinal ganglion cells, how they're going to project differentially to different parts of the brain. And this blue and yellow is this blue and yellow. And this purple and red is this purple and red. This is a really powerful figure. There's a ton of stuff packed into this figure. Can you guys see that now? So like when you read through the book and you're studying through these figures and chapters, that's why I started off the semester saying like, this is going to take you a lot of time each week. Because like when you go through these, really break down what's happening here, and it takes time. But man, are you going to pick up a ton? You're going to soak up so much more information and help to pull together the stuff that you're learning. See what I'm saying? So, and then of course you can see yellow and blue here, right? Yellow and blue. Yellow and blue. So see how this is showing you that different parts of the cortex now are processing different parts of the visual field, which gives us a term that I introduced to some of you guys in biopsych. Do you guys remember that term about a map? Huh?
Topographic.
Close to topographic. Where is this map coming from? Before the LGN. Before that.
I brought it in the slides, but retinotopic?
Yeah, retinotopic. You guys remember that term, retinotopic mapping? No? Okay, so it's the retina has a map of the outside world. Right? You see how this mapping projects onto the retina in a very systematic, organized way? The point of this is to show you that that systematic organization of where things are processed in the retina, what cells across the retina are processing what parts across our visual field, that systematic mapping and representation is maintained as you move through these different layers in the visual processing stream, such that when you look at even the cortex maintains a retinotopic mapping. That is not random happen chance. That is amazing organization in the neurobiology of the visual system. It's really cool. What do you guys think about that? Is it helpful to break it down like that? And we'll see that that's not exclusive division. And the auditory system, we'll see there's something called a tonotopic map, which is a map for different tones. So if you like music, that's really helpful, because it allows you to tell different tones. Even if you don't like music, it allows you to tell different tones of people's voices, so that you can discriminate when you're in the club and somebody comes all dancing up on you, behind you, whispering in your ear sweet nothings. Okay? You can discriminate the tone of that voice and know if that's your boo or if that's somebody that you need to shoot. See what I'm saying? You wouldn't be able to do that if it's all dark in the club and you can't, and it's just a monotone voice like a robot. You need to be able to discriminate those different tones. Yes, Rosie?
I just want to make sure that I'm distinguishing these words right. So topographic, that's just like the general type of mapping within like these systems, but specific to the visual system is retinotopic.
Yes.
Like a subset of topographic?
Yes, but I just, because I'm kind of a word dork, where are you pulling topographic from in this context? Because I think about topography as hiking Mount Baldy or some other mountain, like here in San Bernardino.
So topography is irrelevant. Retinotopic is just like a word you made up. I think is what she's trying to distinguish. Or is that an actual term?
Oh, retinotopic is a big word. It's in the book. It's a big term in the field.
I haven't seen it.
Yeah.
It's in the slide, so that's just why I'm confused.
Yeah.
I just don't know what is specific to topographic. Is it just the projection itself? I can show you this one.
Yeah,
I just don't. I just want to make sure I distinguish them right. Because it's talking about the cortex, so it might just be a different area.
Oh, yeah, so it just means, it just says there's a topographic projection of the retina, which just means that there's areas of the cortex, it means retinotopic. In this case, they're interchangeable. They're interchangeable.
But retinotopic is specific to the visual system.
Yeah. Sorry, I'm a dork. When somebody says topographic, I just think about hiking and mountains, and I can't break that from my brain.
No, you're fine. It just confused me last quarter, and I just want to make sure I don't do the same thing again.
Yeah, and I can't believe if I don't think that there would be a hiccup on that, on like a test or a test question, and if there was, I'd be pretty flexible with it, so. Okay, can we move on? Okay. Okay, so we have that. This, again, another great image, right? So this is not from the text. You guys did a very nice job creating some supplemental views, this is probably nicer than what's in the text. This is a really cool figure, I think. Really useful. Showing these, see how remember we called those optic radiations? The thalamocortical projections from the thalamus. What part of the thalamus? LGM. Is it up there? You guys cheated. Okay. The thalamus projecting here. Now what's happening here, right? There's a lot. See how these extra colors move out? Because they're showing different parts of the visual cortex. So we can really get more granular because this isn't biopsych, right? We're going to get more specific. Okay? So it's not just, oh, one big chunk of the occipital cortex, that's where it is. Okay, gotta go, switch to auditory system now. No. We subdivide this into different areas. So we have V1, which is basically visual cortex, basically the first stop. Then it projects up to the sort of outward. It radiates out to an area around it we'll call V2. And as you might imagine, the next one is what number? Yeah, right? These scientists, they come up with some pretty funky words sometimes, but they also don't like to confuse themselves either. And so sometimes they get pretty helpful and just go 1, 2, 3, and whatnot. So even area 3, you can have 3A, a little bit above it. Okay, you have some other areas, the occipital lobes, V3, V4. Okay, so you have this information. It's going to come here, right? And then it's going to start to spread out depending on what type of information it is. Which in itself is absolutely phenomenal that the brain has that distinguishing ability to do. Right? And that's pretty cool. So anyways, so we got that. How much is left from your guys' group? We're holding back this other group. I want to get to them. We already talked about this stuff. This is the same information. We talked about retinotopic mapping. The scotoma is what we've already talked about, which is that spot where nothing can be perceived. Right? But sometimes you can still test people for their memory for things that might have been shown in that area, and they might perform above chance, which is pretty wild. Again, attesting to the power of the unconscious mind. It's pretty remarkable. We'll talk about non-conscious memory later on in the class, maybe. It's pretty cool. But yeah, so anyways, so we've already talked about this, right? We just mentioned the scotomas, we talked about cortical blind sight, and we talked about hemispatial neglect. So because we got this out of the way already, I'm going to move through that. Ooh, here you go, guys. You should slow me down, tell me you had memes. What you got? Blind sight, I can't see a thing. A real... I don't understand the cartoon. Can somebody help me?
Yeah,
What's that?
I must have heard
Oh, okay. Oh yeah, hemispatial neglect. Remember we talked about that? You can only see part of the world. So this is what you and I see, let's say, when we sit down. When we wear stylishly brown pants. Okay? They can only see one side of their perceptual field. All right, we talked about damage, these clinical situations. Okay? If they're looking on the table, they'll only eat half of their plate because that's what they see. They don't see this part. All right, so next time you're yelling at your kid to eat everything on his plate for dinner, make sure he doesn't have cortical blind sight first. Okay? Because if that's true, then you're picking on a handicapped kid. That's not cool. You know what I'm saying? Like, you got to make sure you're not yelling at him for the wrong reasons. Or her. I don't know if you have a boy or a girl. But anyway, so yeah, it's pretty remarkable. Actually, a friend of mine in graduate school. You want to talk about how big of a dorks we were in grad school? We had a Halloween party, and one of the other graduate students in the neuroscience program, for his costume, he dressed up as a hemispatial neglect patient. And so he shaved half of his face, and he tucked in like half of his shirt, and you know, because that's what they do. I mean, you don't see the other side, and so you know that you only take care of one side. It's just, again, cognitively on that attention is just not there. So pretty fascinating stuff with the visual system. Or if that's my eyeball, okay. Pretty cool. Is this the end of it? It's not clicking forward on me, so I'm assuming it is.
That's the last slide.
Okay, so that's cool. Enough ado about that. Let's get this other group cracking. What slides were we starting on for y'all? 52.
Receptive field.
Yeah, okay, so we talked about this a little bit. Are you up first, Roman?
Yeah.
All right, so the floor is yours. I'll be your fan of white.
Can you actually higher the thing so I can draw?
Oh, yeah, sure.
I'll raise that up.
There you go, sir.
So short definition of the receptive field is basically sensory cells that are stimulated by light, so excitatory or inhibitory, so that'll tie in kind of with the receptive field of on and off center ganglion cells. So think inside the retina, those retinal ganglion cells. So let's say if light's hitting on right here, that'd be an on center. Yeah. So that'll change the polarization. So think of it this way. -70. And then that'll just kind of shoot up. Now if the light wasn't on center, let's say it was over here, keep in mind this is the entire receptive field. This would be,
For what kind of cell, by the way?
I think it's the ganglion.
Yeah, okay, so you're talking about the receptive field for one ganglion cell. So we used to be talking about the receptive field for like the brain or the eyeball, right? We have like zoomed super narrow, and now you guys all know the idea of a receptive field now? But now for one cell. That's why it's just the circle instead of that big, wide, colorful grid.
So this would be considered an on-center. An off-center one would be if the light was hitting on out here. So just assume this is all light right here.
So what's that tube you're drawing?
That's light.
Okay, so we gotta explain that to people, right? Because let's just remind everybody that what he's drawing here, this circle, this is supposed to be the receptive field, and just like a bullseye in a dartboard, it has a bullseye in the middle. There's also the area surrounding the bullseye that we call the surround. We call the center. The center. Seriously, that's the scientific word for it. So what this thing is that he's showing, and what this thing is that he's showing, is just it's just a spotlight. It's just a cartoon of a spotlight. So that's what he's dialing into here as he's explaining this.
So this will be the on-center cell, and this will be the off-center cell. So
how did we note that? If we needed to draw that.
That's the way I know it. Now, essentially, let's
call on center anything that what we mean by on center means that the cell fires when there's light on the center. Okay? Because this stuff is going to sound kind of funky. So let's put a little plus sign on the center so we know that it fires when it's on it. And if it's on in the center, then we put a negative sign in the surround so we know that if there's light, if you shine a light here, is that going to make the cell happy or sad? Sad. That is, it's going to make it less likely to fire. So it's inhibited. And here it's excited. So positive for excitement, negative for inhibited. Now, show us what the off-center cell would look like instead. Yeah. You guys see how that's opposite?
Essentially what's happening is, let's say if it's the on-center cell and light's hitting the center right here, more action potentials are firing. Now, if we brought the light out here, on the outside, action potentials will not be firing as much. Versus where if it's an off-center cell, if it's on the outside, the action potentials will be firing more often, more frequent, whereas if it's on-center, or if the light's hitting the off-center cell on the center, it wouldn't be firing as much.
Now, what's that thing you drew over there with the 70 and it looks like a little car? What's that thing?
That's the bipolar response. bipolar cells responsivity.
Sure, it's the bipolar cells?
Bipolar, yeah, that's what it's saying.
Okay.
right here.
Okay, so
It's
then,
changes in polarization, essentially.
so this is your memory potential. That's
Yes.
your electrical voltage.
Yes,
And what's this line?
that is. I cannot tell you. That's its resting potential. No.
Okay, and this is just showing the change for that.
Yes,
Just showing that this is normal with no light on.
based on the. It'd be more of this way.
Yeah, okay.
So yeah, so light hits, it'll change in the polarization, and let's say the light was taken off, it'll essentially repolarize.
Yep.
And then for the off center, or let's stay on this one real quick, if it went off center, if the light was hitting over here, let's say we're back over here, it wouldn't, what would you call, this isn't it's depolarizing, but it's not like an action.